Polypeptide is a singular linear of amino acids that are linked covalently by polypeptide bonds. These polypeptide chains go on to make proteins essential for growth, repair, function, structure, and regulation of the body's tissues and organs. With this, it is important to understand polypeptide synthesis, which simply means the process of making a polypeptide. It is during this when two main steps occur: transcription and translation. Polypeptide synthesis starts in the nucleus of the cell, the location of the DNA, and the site of transcription, which is the process of the first step. DNA is made of phosphate bonds and a sugar called deoxyribose, which is from the backbones, and then four nitrogen bases with each pair joined with a hydrogen bond. To begin with polypeptide synthesis, the enzymes of helicase unwinds and unzips the portion of the two strands of DNA. One of these strands will be used as a template of DNA replication. This small DNA has a start and stop signal, used in order to recognize the code needed for a particular protein needed at the time. Complementary RNA nucleotides pair with the chosen DNA strand bases. Cytosine and guanine nucleotides pair together, and with adenine and thymine nucleotides pair. However, anytime RNA places a complementary on the DNA, it will place uracil instead of thymine. The RNA polymerase enzymes that binds these RNA nucleotides together, making a new single-strand RNA. This new strand is called the messenger RNA or mRNA. This provides the specific codes and information of the exact order of amino acids that are needed to be bonded together to form a polypeptide. Once formed, the mRNA travels from the cell's nucleus through the nuclear membrane and into cytoplasm, where it will attach to a ribosome made of all RNA or ribosomal RNA. Thus, begins the next step, which is the translation. In the cytoplasm, there are many tRNA molecules available, or also known as transfer RNA, that carry an amino acid. The monomer of polypeptide, the ribosome within the cytoplasm, gives the mRNA code three bases called codons at that time, and adds the complementary anticodon found on one end of the tRNA. These codons provides the information determines the related amino acid. The other end of the tRNA that related amino acid integral for building of polypeptide with the first tRNA and amino acid. The two amino acids carried by tRNA will link together to form by forming a peptide bond. Then the first tRNA and its anticodon will detach, deep in behind the amino acid where it can pick up another amino acid. Attach the next tRNA carrying another amino acid will move in and attach its anticodon to mRNA and his complementary codon. The mRNA will shift through the ribosome and the next molecule can come in. As more tRNA anticodons match with the codons, the amino acid chain becomes longer. This will continue until a stop codon is reached. A codon does not code for an amino acid. Once the stop codon is read, the amino acid chain will detaches and the polypeptide is formed.